Ecology. So what do you understand from microbial ecology? Uh, what is ecology then? Let's start the lesson by asking this question. What is ecology? Anyone?
The studies of organism to its surrounding.
Ah, so ecology comes from the word oikos and logos. Okay, oikos means household, ecosystem, the habitat, whereas logos are laws. Okay, rules. So the rules dictating the household, the rules dictating the environment. Okay, so we are concerned about the environment and the the laws that govern what we see. Okay, for example, uh, from observation, people know that when a habitat is productive, the diversity is higher. The diversity of the organisms are higher. So once they observe the same pattern over many places, then that observation becomes a hypothesis, and when the hypothesis is tested, then it becomes a law, ecological law. For example, growth rates will be higher when temperatures are higher within a certain range. So that also is an ecological law. Okay. Uh, so ecology is a field science. You observe and then you note down your observations and then you analyze your data. So that it's a field science. Unlike laboratory experiments where you can control the variables, where you can test certain hypotheses, ecology, the approach to studying ecology is different. You have to observe, then note down your observations, collect lots of data, then analyze it. So because of this, it doesn't have the robustness of experimental or laboratory setups. So how do you test uh, whether your observations are correct? How do you test whether your hypothesis is correct? So that's why statistics play a very important role in ecology. Okay, so that's why a lot of the biostatistics that we learn actually evolved from uh, ecology. Okay, so ecology is basically that is the definition of ecology. But the title for the course is Microbial Ecology. That means basically we are studying the ecology of microbes. So in your opinion, what are the microbes? Microbes cover what range? Anyone?
Organism that we can't see using naked eye,
Yeah, but give me examples.
such as bacteria, fungi,
Bacteria, fungi. What else? Anyone else?
virus,
Virus, and I think protists.
Okay, protozoa, protists. You have viruses.
Mm-mm.
You have algae, phytoplankton. Okay, so basically we are talking about all the microorganisms, uh, the ecology of the microorganisms. Okay, So when you want to study microbial ecology, what do you think are the challenges as opposed to studying higher animals? I'm sure some of you would have watched documentaries from National Geography where they film the chimpanzees, the baboons, the animals, their their life cycle and all that. So in microbial ecology, can we use the same approach? What makes microbial ecology different, or what makes microbial ecology more challenging than the, the classical ecology of higher animals? Anyone?
I think because we cannot observe them directly in their habitat because we need tools like microscope.
Okay. So once you cannot observe them in their natural habitat, that means you are actually removing them from their natural habitat. Then that also means that what you observe might not be what is real or what is natural. Okay, so that is one point. Okay, it's too small. Okay, it's microscopic. You cannot observe them with ease. What else? You think of the bacteria. The what makes it different from higher animals?
I think since it is really small, so that and also when we isolate them from a place to another place, when we want to culture it to grow them, so I think there's a potential of contamination, which we can't see this contamination in higher animals, but in the bacteria culturation we can see, so that perhaps it's hard for us to observe the real species that we want to observe.
So I see the final point is quite interesting. You cannot really see what you're supposed to observe, because once you culture something, I mean, from bacteriology one, a lot of bacteria cannot be cultured. So, and whether the bacteria grow depends on the type of growth media you are using. Of that means, for example, if you take a nutrient agar or brain heart infusion agar, very rich agar, you take a sample of the soil and you culture them, and you see a bacteria growing. It means nothing because the media is so rich. Any spores in the soil would have germinated, okay, or bacteria that are dormant in the soil would have grown because the media is so rich. That's why culture is not a good approach to microbial ecology. Okay. Another point I take from your statement is they're small. Okay. Then when you see the documentaries from National Geography, you can see when they observe animals, they will talk about how, like when they talk about birds, they'll talk how the feathers are different, how the colors are different and all that. So in higher animals, you are able to observe phenotypic differences even among the animals. But in the bacterial world, in the microbial world, there are not many phenotypic characteristics that you can see. Okay, most of the time it's all nearly uniform, where the cocci, bacilli, okay? Because normal staining will not show flagella. So when you do a gram staining, that's all you see, either cocci or bacilli, or maybe sometimes spirillum. But that's all. You don't see any other characteristics. Okay, then there's no other phenotypic characteristics that you can see. So that is another challenge. So here we have covered three challenges. One is they're small, you need special tools. Two is phenotypically they lack diversity, so you can't really differentiate them just from their phenotype. And then the third one is once you culture them out, they changed. Okay, well, I mean the culture method is not really feasible for microbial ecology. Okay, anyone else with comments? Okay, no comments. Okay, so we've discussed the term ecology, okay, where ecology means the laws governing the household, where the approach to ecology is basically based on observations and patterns, and then you come up with ecological laws. But for microbial ecology, where we study microbes like fungi, virus, algae, bacteria, and protozoa, microbes are minute, they are morphologically uniform, and when you remove them from the habitat, they change. So these are challenges in microbial ecology. Okay. So let's go back to the meaning of ecology, where we say we are looking at, we're trying to find the laws governing the household. So basically, microbial ecology means we are looking for patterns and trends and laws that will affect, that will influence microbial community. Okay, so we can generally separate these. So we are looking at factors that can affect microbial ecology, microbes. Okay, so in this course, we are focusing on factors that can affect microbial abundance, factors that can affect microbial diversity, and factors that can affect microbial process rates. Okay, so microbial abundance, diversity, and process rates. So if you look through your lecture notes, you'll see that we focus initially on abundance, then diversity, and then process rates. So let's just talk about microbial abundance first. So what are the factors that do you think affect microbial abundance? So microbial abundance, okay, first, how do you think we should measure microbial abundance?
We determine the community.
No, yeah, like we're talking about abundance. So if I ask you to determine the microbial abundance in Lake, what will you do?
extract the sample first.
What will you do? You can take the sample, okay? Take the sample of water, then what do you do with it?
Serial dilution and find the CFU per mil.
Okay. So is that RD? Was that RD just now? Yes. Okay.
Side.
Side. Okay. Sorry, I didn't see it.
It's okay.
Okay. So side is suggesting culture method. But I think Nurul just now said once you culture them, once you remove, you are already removing them from the habitat, and what you're observing is not necessarily the truth. And moreover, we all know that culture only isolates up to maybe 5% of the population. So if I ask you to check the microbial abundance in Lake, then you know that microbial culturable method will not work. It's not feasible, right?
Yeah. So you do direct methods instead,
Yes, you can do direct count.
and then estimate the relation.
What kind of direct count methods do you know that you have learned that you can still remember?
The hemocytometer.
Yes, hemocytometer is feasible. You take a sample of the Lake water, put it in hemocytometer, and you observe. Then you can count within the grid. That's okay. I accept. But of course, the fundamental flaw weakness in hemocytometer is that it is harder to view. So now we have a epifluorescence microscope that we can use to stain the bacteria, the total bacteria, and then to view it under microscope. Using a fluorescence microscope, we can see the bacteria clearly. Okay, so that is one approach, direct count. But so does that mean culturable method cannot be used at all?
Can, but we use it when we want to find the abundance of some specific species
Oh,
of bacteria.
yes, very good. For example, in water microbiology, there's a lot of emphasis on fecal pollution indicator. There are bacteria that is used to indicate pollution, fecal pollution. Fecal is feces, so there are bacterial indicators for that. The most common ones is E. coli, Escherichia coli. So if the question asks you to enumerate or tell them the abundance of E. coli in Lake, then yes, you can use culture method. No problem at all. Okay. But if you're looking for total microbial abundance or this microbial abundance alone, you cannot use culturable method. You have to use direct counts. Okay. So okay.
Okay, what about the spectrophotometer? You measure the light intensity.
Oh, when you do spectrophotometer,
Now, is that not feasible also?
you are measuring absorbance and then converting the absorbance into CFU per mil. Yes?
Mm-hmm.
Okay. So the first question is, what model organism do you use to convert the absorbance to CFU? Then the next question will be, when you sample Lake, not only the microorganism will give you absorbance, there are other particles in the lake water that will also absorb light. For example, the algae, the detritus, the particulates, the suspended silts and solids. Okay, so you it will not be accurate, and usually people do not use it.
Right.
People use spectrophotometric method when you're doing lab experiments, when you're working with pure culture, when you can control the broth, when you can control the type of culture you're enumerating, then easily you can use spectrophotometer, no problem. But when you are working with environmental samples, then it is not feasible to use spectrophotometric methods. Okay,
Right.
but all these methods, that's why you must, whenever you learn a new method, whenever you come across a new method, you must see you must know their advantages, their disadvantages, and what they are good for, in what occasions you can use them. So once you have a good grasp of all these factors, then you will become a very better scientist, because you will know when sometimes what kind of approach to use and what not to use. Okay.
Okay.
Okay, so now let's just say we settle we will look at the microbial abundance. Let's just say we will use the simpler method. We'll use a hemocytometer. Okay. So the next question I'll ask is, so I'm asking you to tell me, to enumerate the microbial abundance in Lake. So we have decided that culturable method is not that good. We'll use direct count methods. Direct count methods can be hemocytometer, can be epifluorescence direct counting. There are also other methods, more advanced. For example, flow cytometry. Okay, that one you will probably cover in your instrumentation. Okay, so now you have your approach, you have your method. So how are you going to do this? Is it straightforward? Anyone? So you already have your method. So do you sample once, twice, 3 times, or once is enough? So you are asked to measure microbial abundance in Lake. So I'll guide the question here. So how many times sampling do you think is enough?
I assume quite a lot.
Quite a lot.
Yeah, maybe like at least 5.
5. Is 5 a lot?
And I also think that you have to take from different places as well.
Ah.
Yeah,
So
like different
site
sections.
site has initiated a new point. Different site. Okay. So when you recommended, or when you suggested different sites, that means you have a feeling that if you only sample from one place, it might not be accurate, right? So does that mean you have a feeling that uh, other places will maybe have more bacteria. Okay. Is that why you suggested several sites? To give a better average?
Yeah, pretty much.
So here we're looking at spatial heterogeneity. Heterogeneous. Okay. Homogeneous means well mixed. Okay. So you realize that they are not necessarily well mixed. Okay. Because if you notice, maybe the bacterial count will be higher where there is the the water treatment system, where they have the plants, the plants for bioremediation. Okay. Also, basically, when the drain flows into the lake, that may be a place where you have higher bacterial abundance, microbial abundance. So when we talk about spatial heterogeneity, do we only talk about the surface water? What about the bottom? What about bottom of the lake? Will it be different?
Yep.
Yes.
So when you look at spatial, you have to be aware that you're not only talking about surface, you're also talking about depth. Okay? But luckily UM Lake is not that deep. Okay? So but so if you want to have a good average, probably you should take one at the surface and one at the bottom, just to give you a average. Okay. What about you have here spatial heterogeneity? Okay. So you're talking about in terms of space, that there is heterogeneous. What about time then? Is that temporal heterogeneity?
Probably because some microbes are more sensitive to light.
Ah, that's good. So, because phytoplankton carries out algae phytoplankton carries out photosynthesis. Okay, and photosynthesis is dependent upon light. So they will be more active. They will be reproducing. They will be carrying out more processes when there is light during daytime. You also know that photosynthesis fixes carbon, produces sugar. That means when the phytoplankton is active, the bacteria will also be more active because more food from the phytoplankton, right? Because phytoplankton is just like the trees, they are the primary producers in aquatic environments. So when the primary producers are active, that means the heterotrophs, the bacteria, will also be active. So there is temporal heterogeneity. So now you know there's temporal heterogeneity between daylight and nighttime. You know that there's probably spatial heterogeneity, top, bottom, and also maybe near the inlet, away from the inlet, and all that. So that's why when we talk about sampling a lake, there are several approaches you can do. One is you can do a linear transect. So at every certain distance, you sample it out. Okay. to give you an average. But this is called linear transects. Okay. But other than linear transects, there are also grid sampling, which means you sample at every grid, every intersection. Okay. So that will be called okay. Other than this, there is also the easiest one probably, random sampling. What you can do is, what you should do is actually do a grid, assign a number for each grid, or just throw a paper or throw a dice somewhere and see where it lands, and that's where you sample. Or yeah, that's random sampling. So we're talking about measuring microbial abundance, the approach to take into consideration whether there is spatial heterogeneity, whether there's temporal heterogeneity, and then using different kinds of sampling transects, grid or random, to pinpoint your station. Okay. So when you talk about temporal heterogeneity, we have covered the day and night, daytime and nighttime. Do you think there are other factors that can affect temporal heterogeneity?
Temperature.
Temperature is very important for biological processes. In Malaysia, our temperature varies within a day, maybe from 25 to 35. At least nowadays it's down to 35. So yes, temperature, but then most probably temperature also relates to day and night, because temperatures will be warmer during day and colder during night. Okay. However, temperature is very important in temperate countries. Okay. An example of a temperate country?
The UK.
Are you familiar with the word temperate?
Yeah, 4 seasons.
Oh, okay. So example will be Korea.
Japan.
Yeah, nice. Japan. We are
We are tropical.
okay. So tropical, characterized by 12-hour day, 12-hour night on average. Temperate, no. Temperate, it depends on the season. Okay. So the daytime can be as long as 16 hours a day to as short as 8 hours a day. Okay, so it can vary. And temperatures also can vary over a wide range. In the winters, it can go down to below zero, especially in Hokkaido, Northern Island. Okay, so in the summer, it can go up to 30. Okay, so we're talking about more than 30 degrees Celsius range. Definitely temperature, definitely the seasons affect. Okay, the seasons play a important role for temporal heterogeneity. But in Malaysia, not so much because we are tropical. So temperature, not so much. But other than temperature, what else do you think affects the temporal heterogeneity in our country?
The weather, maybe.
More specific will be the monsoons. Okay, where when we have the dry versus the wet monsoon. So this will definitely affect your sampling. Okay, because dry and wet conditions will affect the lake a lot. Dry conditions, lake will have less water, so conditions will be different as opposed to wet conditions. A wet monsoon, when the volume of the water in the lake increases tremendously. Okay, so it affects. So yeah, so you can say it's the weather. Okay, it's more related to the rain. Okay, so we are talking about sampling. No, measuring microbial abundance. So we decided to do direct count. Then we've considered the spatial heterogeneity. We've considered the temporal heterogeneity. We've considered the approaches to selecting our site selection. Okay. But we still haven't decided how many samples to take. So how many samples do you want to take? How many samples and how many sampling trips? Because the number of samples will depend on the type of sampling approach you use, whether random, linear, or grid, or whether you do surface or bottom. Okay. But how many times, how many trips to go to the lake will depend more on whether you are covering temporal heterogeneity. So in order to account for day and night differences, what do? Sample. Anyone? You do two trips? One in the daytime, one in the nighttime?
Yep.
Then what about this? Dry and wet. You do two trips? One during dry, one during wet?
Maybe.
So let's just say you do. That means how many sampling trips are there then?
Four.
Okay, 4. But you're only talking about one day, one night, one dry, one wet, right? Is that enough? One dry and one wet. This one side say 5, right? You need 5, right?
Yeah. At least.
At least 5, Okay.
For each lah, I mean.
Yeah. So when you talk about number of sampling trips, the first consideration is this. Okay. Sampling trips are expensive. Not to Lake, of course. but if you're doing sampling outside, okay, planning a trip out of the state or to the seas can be costly. So you cannot be over ambitious. Okay. Minimum replication best is 3. Okay, n equals 3. Why not 2? Why not 1?
Two less.
Uh,
1 is too little, and 2, you might as well go for 3.
can I rub this? Yes, okay.
Yeah.
Do you have any questions about this? Especially the quiet ones. Do you understand?
Yes.
Yes.
Okay. Okay, let's just say if you do one sampling, you get a result here, right? Let's just say you get a result here. So you will report this as the abundance is like this, okay? No, this. Maybe you're not so confident whether that's the result. So you do again. Then you realize that it's here. So how? You take the average?
Yes.
But then let's just say you're still not sure. You're not confident, you go again. Then you realize the third one is here. So do you still take the average?
Think so, yeah.
You have to, right?
No, because the central tendency is towards here. So why do you take this average? Why do you consider this when most of the points are here? That's why minimum replicate should be 3 in order for you to detect 4. Because field science is different. When you are measuring from the environment is very variable. It's not like laboratory experiments where you can control a lot of the conditions. When you're doing field science, a lot of things you cannot control at all. So what we usually do in the lab is we will measure definitely more times. The more the better, because if you realize, if you understand, I mean, if you remember for t-tests, I mean, in ecology, statistics is important, so you need to use statistics. So when your sample size is small, you use t-test, but t-test is better when your counts are around 30. So if you are having dry season and wet season, maybe we'll do 6, 6. Then day and night, there'll be 12, 12. Then we'll get a total of 24. And then when you use statistical testing, your results will be, you have more confidence in your results, more confidence in your results. Okay? But when you do 3 or when you do 5, of course you can still do t-test or ANOVA or whatever test you want, but your results will not be as robust. Once you do testing, statistical testing, there's a big possibility that you will not get any results. You will not be able to test your hypothesis. Okay? So the so the, a better way of doing this will be probably to set your hypothesis first, and then to determine your experimental setup, and then to see your statistical testing. Okay. That means you have a question, research question, then you determine a hypothesis that is testable, that you feel you can do. Maybe your hypothesis can be microbial abundance differs between dry and wet seasons. Okay, that's a valid hypothesis. That one can be tested. So experimental setup means, since you want to test dry and wet season, you want to check the microbial abundance. So you will set the method. We'll be fixing with direct count. Then we'll set the sampling sites, how many stations to take so that we cover the lake properly. And then we'll set the sampling period, 6 times in the dry, 6 times in the wet, so that we can compare and test the hypothesis. Okay, so since we are doing testing between dry and wet, we are testing 2 variables, 2 conditions, so we'll use a t-test. And then once you've done all this, of course, before you do hypothesis, you also have to do literature review. Why do we do literature review? Why is literature review important? We keep asking the students to do literature review. What is literature review? Anyone?
Survey.
A survey of what? Of the things published.
On the experiment that we want to help.
Oh. You do literature review to know a historical background, the background of the research that you are interested in, so that you do not repeat what has already been done. You do not repeat other people's mistakes, and you proceed on from other people's developments, which means people have done a certain stage, but there are still questions that are left unanswered, so you progress upon them, progress upon their work. Okay, that's why literature review is important, because we don't want the students to be repeating what has already been done. Because in UM, especially for postgraduate studies, you're supposed to publish your work. If you're repeating what is already done, what has already been done, no one will publish your work. So you'll be stuck. Okay? That's why literature review is important. So that you know the state of science for the research that you're interested in, and you can progress upon it and not repeat what has already been done. So once you've done the literature review, you're able to come up with a hypothesis. You found your suitable experimental setup and suitable statistical tests to test your setup. So once you've done your results, once you obtain your results, you'll have your wet, your dry. Then you do the testing, and then finally you reach a conclusion whether to accept or reject your hypothesis. Okay, so that is what you should be doing for any scientific research. Okay. Okay. Any other questions?
No, so far.
Okay. So
Doctor, sorry. What if the conclusion does not support the hypothesis? Is that a waste of experiment time, or like is that common in science?
oh, site is it?
Yeah.
So you're asking like the panel for the grant evaluators whether it's a waste of time or waste of money to do these things. That's why nowadays, even in Malaysia, all the grants, they're always looking for this. Return on investment. So if the government gives you 100,000 ringgit to do research, they want to know what can they get back. So in order not for it to be a waste of time, you have to do your literature review properly. Then usually for me, I will probably do a which means using whatever leftover grant that I have, I will carry out a small scale preliminary investigation of what I want to do next to see whether it's feasible. Then if I feel it's feasible, then I just apply for a bigger grant.
Yep.
Okay. And the good thing about this is you can actually put the results of the preliminary investigation into your grant proposal. Okay, it's actually better, then the evaluators will see and will actually be more confident with your research proposal. Okay, but yeah, that's a valid question. You see, once you set your hypothesis, if your results don't prove your hypothesis, then your hypothesis is rejected. Then that's it. It's rejected. Sometimes it's like that. Not everything has to be positive. Not every result have to be positive. Okay, sometimes it's like that, negative. But still, you still have to do it. Someone has to do it. If not, no one will know. Right?
Yep.
Okay, so we've done. Okay. Oh, so we took so much just to do so little. Oh, okay. Okay, so before we proceed on the before we proceed looking at microbial abundance, let's talk about a bit about history. I think history is everyone's favorite subject, right?
RD's favorite subject.
Okay, I thought it's everyone's favorite subject. Okay, microbiology. When you talk about history, who do you think of?
Robert Hooke.
Robert Hooke.
Anthony
Wow.
van Leeuwenhoek.
Pardon?
Leeuwenhoek. Anthony van Leeuwenhoek.
Wow. You're all rare, y'all. Anyone else? So Robert Hooke, Anthony Van Leeuwenhoek, that's the microscope and the okay. What else? Give me something more recent.
Pasteur.
Pasture, yes. That's the one I want. Who else? What's the full name? Pasture. You all know?
Louis Pasteur.
How do you all spell? Okay. Where is he from? Where is he from?
For France.
Good guess. Who else is famous? Pioneers? Microbiologists? Our future budding microbiologists? Come on. How come Leon is so quiet? Anyone? Hello? Hello?
How about Edward Jenner?
Pardon?
Edward Jenner.
Oh yeah, vaccination, yes. But no, I'm looking at this
Yep.
guy. You know him?
Yes, the cock pistolette.
Yes.
From
Robert
where?
Britain.
Okay. Oh yeah, what y'all covered this right in what the general microbiology, right? Do y'all cover this?
Yep.
Okay. So what so I consider this guy to be the father of medical microbiology. So this guy to be the father of microbiology. Okay. So they make tremendous contributions to the field of microbiology. Louis Pasteur was the one that debunked the theory of spontaneous generation. How did he do that? How did Louis Pasteur debunk debunk or the theory of spontaneous generation?
The what we call swan neck.
Yes. Who's that?
Nuril.
Oh, Nurul has good memory. So how does the swan neck works anyway?
because when he put the water droplets inside this one glass at the middle, so that he know that no other microorganism can enter the flask, the tube, so that and after he observed after a few days, and it shown that no other living things in the broth.
Yeah. So the theory of spontaneous generation was based on the observation that meat can go rotten with flies and maggots over time. So they say, oh, spontaneously they generate. So in order to debunk it, you have to. So what they did. So Tyndall started the experiments. He boiled the rich broth and showed that once you boil it, nothing grows. You know, it won't get contaminated. You won't. But then the the criticism of the experiment was that there was no air. Okay, no air. They said spontaneous generation requires air. When you boil it and then you keep it in airtight containers, of course you cannot see any spontaneous generation. That was what they criticized the experiment. That's why pasture this one was important. With this, air continues to travel in, but the broth remains uncontaminated. That means there is no such thing as spontaneous generation. That's why it's important. Okay? If not for this, we will still be thinking that there is spontaneous generation everywhere. Okay? So only after you debunk the theory of spontaneous generation can you focus on the fact that there are microbes, that microbes exist, and that microbes can cause diseases. So initially, at that time, the focus was always on diseases. Okay, why diseases? Because it's important, and diseases not only for humans but also for livestock. Okay, so Louis Pasteur helped a lot, especially for the farmers, for the wine industry with his pasteurization. Uh, he helped with the sheep farmers, the poultry farmers. Okay, by by looking at problems from a disease point of view, which means when he sees animals dying, it's not just because of some witchery or some bad air or anything, but he says there's probably a microorganism involved. So then they would proceed to do experiments to find that microorganism, to culture it, and then even found a vaccine for the uh, Pasteurella multocida, the the cholera in fowls, in poultry. Okay, so that's why he's important. Okay, then Robert Koch, of course, from his lab, the modern techniques that we are using, the petri dish, the agar, the isolation, the culture techniques, all originated from his lab. Okay, not him alone, but sometimes people work in his lab. They bring in ideas like the agar, the use of petri dishes. So, but because his lab was the main pioneer microbiology lab, then other people go and learn from him, from his lab. That's where most of the techniques that we have now originated from. So, father of medical microbiology with his Koch postulates. We have Louis Pasteur, father of microbiology. So, when you you need to know history in order to appreciate the field of microbiology that you are in. Okay? The tremendous achievements in the late 1800s and the early 1900s. Okay, most of the Nobel Prize winners in medicine were in the field of microbiology. Okay, at that time, there were a lot of diseases that people don't understand, but because of microbiology, we are able to determine, okay, the cause of diseases. And only then, only once we accept that diseases are caused by microbes, that we are able to treat it properly. Okay, if not, we'll be yeah. So if you remember from our previous lecture on bacteriology, only after we realize microbes are involved, then with antibiotics, with vaccinations, you see the rate of mortality decreasing tremendously. Okay? If not, we'll still be, uh, if not, the mortality rate among children will still be very high. So tremendous achievements and contribution to the world. Okay, so Louis Pasteur, you know, you should read their autobiography so that you appreciate what they've done. So he he found the pasteurization step. He didn't patent it. He gave it freely. So what do you know of any famous sayings from Louis Pasteur?
No.
I teach you some lah, okay? Okay? The role of the infinitely small is infinitely large. Okay? Very true. Okay? The role of the bacteria, the role of the viruses is very, very important. I mean, if a virus can if a virus can stop the whole world from functioning for 1 year, isn't it incredible, Yeah, COVID-19 put a actually like put a break on everything. So yeah, so this is interesting. Okay, the role of the infinitely small is infinitely large. Okay, so that's why you're doing microbiology, because you know this, right? You know that the role of the infinitely small is infinitely large, right?
Yep.
Yes, that's why you're doing microbiology. So we know Pasteur, we know Koch. So, but their approach is culture, okay? Their approach is culture, 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 culture. But culture doesn't work for ecology. So for microbial ecology, you need a different approach, okay? But their mindset is too fixed on culture. When their focus at that time was mainly on diseases. They were not wavering. They were not doing other stuff. They were only focused on diseases. So you need other people to come in, and so you have this. Okay. Sergei Vinogradsky from Russia. So you know culture is not uh, so good to study ecology. So you need a different approach. So what Sergei Vinogradsky did was very interesting. He had columns where he put soil mixed with water, and then he just placed under the sun day and night to see what happens. Now, of course, when you have a column like this, you have a natural gradient of oxygen, where the oxygen is high on top and decreases to the bottom. So from here, you'll see algae growing here on top. Then you'll have maybe photosynthetic bacteria here. And then around here, the anaerobes will come in and other organisms will come in. So you see layers of microbial population. So we know, so they call this the Vinogradsky column. So this is a different approach. No culture is involved. What happens is once they can observe the microbial population, because some of them will have color, you'll just remove them and look under the microscope to see what the morphology of it and all that. Okay. Then the experiment that he does was he put certain elements into the soil, maybe sulfate or maybe carbonate. Okay. So when he put sulfate in, he realized more hydrogen sulfide producing organisms uh, are present. So he is able to study chemolithotrophy. From this Vinogradsky column by manipulating the chemicals here. Okay. So his achievements are important for microbial ecology because without a different approach, we will never be able to proceed further. Okay, that's why this is an important achievement. So he's considered the father of soil microbiology. Okay. Okay. Any questions? Anyone?
No.
Nope.
Okay. So the next one that I want you to know is this guy. This is the last scientist that I'm gonna share with you. Martinus Buyerink. He's Dutch. Have you heard of him?
Yes, yesterday.
Yes.
Huh? You heard of him, in virology?
Yeah, virology.
Okay. So tobacco mosaic virus filter. In the field of microbial ecology, he's the one that uh, uses a different approach. Because as we mentioned earlier, culture is one way, but it is not entirely suitable for microbial ecology. So you need different approaches. So Sergei Vinogradsky came up with his Vinogradsky column, where he was looking at the whole microbial community, the development of the whole microbial community along a certain dissolved oxygen gradient, and he he was able to see how the microbial community changes when he changed the elements in soil. Okay, when he added certain chemicals to the soil. For Martinez Bioring, he uses uh, he recommended the use of enrichment culture. So the most famous experiment was probably the one where he has N-free media. So N-free media means nitrogen free. The medium has no nitrogen at all. So what does that mean? So if you inoculate the broth with soil, a little bit of soil, then after 2 weeks you notice growth, things growing. So what conclusion can you make from this?
The bacteria doesn't need nitrogen.
But you know all life requires nitrogen, right? For DNA?
Oh, the bacteria is nitrogen fixation. Fix.
Yes! If the bacteria can grow here, even in an N-free media, that means the bacteria fixes nitrogen. Where does the nitrogen come from then? If there's no nitrogen here?
Air.
Yes! Air, 70% or 71%, I'm not sure. Okay, so that is a new approach, a different approach than from culture. Understand? Previous culture method was always like you put everything there and then you culture it and then you make sure the media has lots of nutrients, all the suitable supplements are there so that the bacteria can grow. But no, for this it's different. We remove certain things so that we can enrich what we want. So the phrase okay, everything is everywhere. The environment selects. So the microbes are everywhere. It is the environment that selects for the nitrogen fixers. Okay? Any questions?
Did he use the same method for the phosphorus-free medium?
Did he do
So
phosphorus-free medium?
perhaps
No.
maybe at the elements-free medium.
Uh, this experiment was also kind of lucky because there are nitrogen fixing bacteria. Okay. Of course, if you want to, you can also get autotrophs, which means your media will be totally free of carbon. But you place under light, and then you will be able to get algae or cyanobacteria growing, because only the microbes that can photosynthesize, can produce their own carbon, will be able to grow. Okay, so that's another example of how everything is everywhere. The environment will select.